Hello, and welcome to Field Lab Earth, the podcast that's all about past and present advances in the fields of agronomic, crop, soil, and environmental sciences. Today we'll be talking to Dr. Ryan Stewart about how we define soil health. What do we mean by the term soil health? What are the ways in which we quantify it? And which of those ways are actually the most useful? Answers to all these and more coming right up. I'm your host, Abby Morrison. Let's talk about science. Hi everyone, welcome to the show. Today we have Ryan Stewart with us. Ryan is an assistant professor in the School of Plant and Environmental Sciences at Virginia Tech. Prior to arriving at Virginia Tech in 2013, he obtained his bachelor's degree in mechanical engineering from Cal Poly San Luis Obispo and his master's and PhD degrees in water resources engineering from Oregon State University. Prior to starting graduate school, Ryan and his wife served as Peace Corps volunteers in Bolivia, where he worked on basic sanitation issues. Ryan currently leads the Critical Zone Lab at Virginia Tech, which focuses on predicting the movement of water, nutrients, and contaminants by measuring and modeling soil properties and hydrological processes. His group works in various systems, from forested headwater catchments to urban soils to agricultural fields. Ryan teaches classes on pollution science and on soil physical and hydrological properties, including soil health. Welcome to the show, Ryan. How are you doing today?
I'm doing great. Thanks. How are you?
I'm doing super. Thanks for being on the show.
My pleasure.
So why don't we just start with a definition of soil health? What does that mean?
That's a great question to start with. It's probably the $1 million question or the $100 million question. But I'm going to go with the definition that soil health is this representation of the ability of a soil to sustain agricultural productivity as well as ecosystem functioning. And really that's going to integrate together different physical, chemical, and biological characteristics of soil together, kind of finding this optimum sweet spot where the soil is really well-maintained, well-balanced, well-functioning.
Sure. And then there's kind of four other terms we'll just cover real quick to give people some context. So those are cover and cash crops, and then conservation and no tillage.
Sure. So one of the ways that people can improve soil health or maintain soil health is what's known as conservation agriculture. And so some of the things you mentioned there are different variations of conservation agriculture. So cover crops are going to be crops that are planted during times when the soils are otherwise going to be left bare or fallow. And oftentimes these cover crops are not sold for profit in contrast to what we'd call a cash crop, something like corn or soybeans or even in other places, coffee beans, things like that. But these cover crops are used to provide other benefits to the soil. And some of those benefits could be scavenging up those extra nutrients that are left around after the cash crop's been taken out of the field, or providing a cover for the soil so that there's less erosion and less raindrops, impacting right onto that soil surface. Conservation tillage is another form of this conservation agriculture, and the idea here is to try to minimize the soil disturbance. And so one of the definitions of conservation tillage is that at least 30% of the soil surface is left intact and undisturbed and covered with residue from that previous harvest. And then a type of conservation tillage is no-till. And basically the idea here is to really try to minimize surface disturbance to almost nothing.
So now that we've got a base for our knowledge for this, the main challenge that you were facing in this paper is to really figure out how we know if soil is healthy. So why is this such a hard problem and such a difficult thing to define?
Well, so it kind of goes back to that earlier definition that it's not just one aspect of soil. It really is this kind of optimization or balance between the physical, chemical, and biological characteristics and functioning of soils. And so we can come up with different methods and measurements that can measure any of these, physical properties independently or chemical properties. But there's a lot of uncertainty and lack of knowledge on how to actually take these individual measurements and say something more holistically about the health of a soil. An analogy I might go to is you could imagine that you go to the doctor's office for a checkup and they measure your blood pressure and your heart rate, your weight, your height, your BMI. And then you get to the end of your checkup, and the doctor says, well, here's some numbers we measured, but we can't really tell you much about your health, right? We don't have any way to interpret that. In many ways, it's kind of where we are in soil health, that we know how to measure a lot of things, but we don't necessarily know how to say exactly how healthy or unhealthy a given soil is.
And for this research, you were looking at a wide range of these different metrics. Can you give some examples of some of the things that you were looking at?
Sure. So I would say one of the main questions that kind of got us started off was when you go to the literature, when you start looking at what people are doing, you start seeing all these different measurements and metrics that come up. And so that got us kind of scratching our head and say, what are people actually measuring? It's kind of the first step is to figure out what other people are doing. And so we went through the literature and we found up to 42 different methods and different indicators that people were reporting on soil health. And included in that are some grouping together of common indicators, or maybe not common indicators, but various biological microbial assays, for example. We grouped them together as one category, and we still ended up with more than 40 different indicators. So some of the specific ones that we saw were things like soil texture, which just tells you what percentage of different size particles are there, how much clay, how much silt, how much sand. You also saw a lot of studies reporting how much organic carbon is in the soil. But then we got to some less common indicators, such as different enzymatic assays, where you're looking at enzymes that are being produced by the biology in the soil.
So with all of these different ways to describe soil, you had mentioned that you had grouped some of them together if they were really similar, but did you also have broader categories that you were able to group these different attributes in?
Yeah, that's right. So we actually were able to divide these 42 different indicators or sets of indicators into 6 broad categories. And so one of those was physical properties. An example of that would be bulk density, which basically is how much dry mass a soil has for a given volume. There are also chemical characteristics, such as how much phosphorus is available in the soil or what the soil pH is. There are a number of biological indicators, and one of those could be what's the biomass associated with all those microbes that are in the soil. And then another one we called environmental states and fluxes, and this could be greenhouse gases that are coming out of the soil, or even the soil temperature or how wet it is at the time of a measurement. There are also agronomic responses, and of course a very important one of those is how much yield are you getting out of your cash crop, because that's oftentimes ultimately what the farmers cares about, right? I mean, not the only thing, but it's a economic reality that farmers have to have good yields. And then we grouped everything else as kind of general or summation indicators. And so an example of that could be what type of ecosystem services are being provided by an intervention like cover cropping, or to what degree is that ecosystem service, or what amount of ecosystem service is being provided.
Yeah, that's super neat. What I like about this, or I guess what maybe is a challenging aspect of it, is that I imagine that for different types of soils, these different qualities would, like what the standard is for health, would change according to the kind of soil. Like if you're looking at, really sandy soil in Nevada or something, that's going to be very different than a rainforest or a swamp kind of thing. And obviously you're looking more at the agricultural fields, but even within those, I imagine there's a lot of variation.
Absolutely. And I think that's one of those areas of future research that's really important is trying to say, now that we know what to measure and how to measure it, how do we start interpreting these values? Because you're absolutely right. If you went to the beach and took a handful of beach sand, you would get very different values than if you went to Iowa and you took a sample of a farm soil there that's probably got a loam texture, got a lot of organic matter. And so we could do the same exact measurements on these different soils and get numbers, but may not be much meaning, without kind of considering the relative differences between those soils.
Yeah, that kind of brings it back to your doctor analogy of, what might be healthy for one person might not be healthy for another. So once you had figured out the general buckets of information that you were looking at, the next step was really to look at the data to start figuring out some of that context of what might be a more useful test to run, what might have more weight in looking at soil health. So can you describe how you actually went about figuring out these next steps?
Yeah, absolutely. So, and maybe I should say, we went to try to answer this question of what people measured. We went and compiled several hundred studies that focused on cover cropping, like I talked about before, as well as no-till. And so from all of those studies, around 200 in total, we then focused on those cover cropping studies. And a specific question we asked was, which of these 42 different indicators or sets of indicators show some kind of change relative to a control? In other words, a field or plot that doesn't have cover crops. And we specifically looked at a fairly short time scale, after 1 to 3 years of cover crop intervention or inclusion of cover crops. There were a couple reasons why we focus on this 1 to 3 year time interval. For one, a lot of studies are funded for 3 years. There's just a lot more information on these short-term, kind of 1 to 3 year responses. But this is also the time scale of a typical crop rotation. And I also think it's probably a time scale where a farmer is going to want to see some benefit from, going to the extra effort of putting in a cover crop. And so to answer this question, we use what are known as a meta-analysis, where basically we collected the data from these, in this case, 86 different publications, and basically, quantified all the results from those studies. And that allowed us to then compare every indicator reported in those studies for the cover crop treatments versus the no cover crop controls. And so we said basically our threshold was if you see a 10% or greater change in the parameter value in the cover crop plots versus the no cover crop controls, we consider that to be a responsive indicator.
Yeah, and you used a really neat tool, I guess. It's called a research landscape tool to gather all that information for you. Can you describe a little bit about that?
Sure. There's an organization called the Soil Health Institute here in the United States, and one of the tools that they've developed is that research landscape tool. And it's really neat because it basically takes all these different references from peer-reviewed journal articles as well as presentations and student thesis and dissertations and puts it together into this searchable tool. And so you can go to this research landscape tool and you could look for specific problems. For example, you could say, there's too much erosion happening in my field, and you could use that to try to indicate studies that have looked at this problem of erosion. You could also look at the type of soil health indicator. You could go to look at physical properties and look at studies that reported this. And from there, you could sort these results by region, by the type of agricultural system, even by the type of crop that's being grown in your system. So it's a really powerful and neat tool that allows you to really sort through all of this great work that's being done. And I also say that tool is continually being updated, which is even better. So there's more and more resources being added all the time.
That's amazing. I think what's cool about that is you had to be really careful with the keywords that you were looking for, because depending on what you searched on, that could have had a huge impact on your outcome of the data that you found. So can you just tell me a little bit about how you decided what to search on and what to ask this tool?
Yeah, so that is one of the challenges for sure, because this research landscape tool has so many references and so many resources. We had to be careful about kind of narrowing down our question. And that's one of the reasons why we focused on this cover cropping question. And so at the time we did our analysis, we were fortunate in the sense that we ended up with 86 peer-reviewed studies that we were able to pull from. But today I went and looked at the research landscape tool, and it's up to 626 different references and cover crops now. So again, it continues to grow that list of things. So we would have a lot more data to choose from, but there'd also be a lot more effort on our end to try to do that same analysis today by looking at all the cover crop related research that's compiled there in the research landscape tool.
That's amazing. That's really exciting. Hi everyone, I hope you're enjoying the show. Interested in learning more? Ryan's article, What We Talk About When We Talk About Soil Health, published in Agricultural and Environmental Letters, is always freely available. You can find a link to it in our show notes. Let's get back to the show. And once you had looked at all of these different things, what did you find? What were the results?
So, again, going back to our first question of what people measure, and that's why I decided to call the paper What We Talk About When We Talk About Soil Health, is I was just curious, what are people measuring and how did this maybe change through time? And so we looked at how many soil health indicators are people reporting through time. And so we found studies that began in the 1940s and really started picking up in the 1970s all the way through 2017. And one of the things that surprised us was even though, new methods and new technologies come online all the time, the number of soil health indicators that were being reported in these studies didn't really change through time. In 1970, it was about 4 indicators were reported for each cover crop study. And in 2015, it was about 4 indicators per cover crop study. Another thing we looked at was how often were these different parameters being reported. And so as I said before, we had 42 different indicators or sets of indicators. And what we found was only 8 indicators were reported in more than 20% of studies, meaning that only 8 of those 42 indicators were being reported more often than 1 in 5 studies. And so we were a little bit surprised that even with all these indicators and even with all this interest on soil health and cover crops, so few indicators are being consistently reported. Beyond looking at what people were reporting, we also looked at how people were actually collecting their data. And so one of the other questions we had was, what kind of soil samples are people collecting when they're talking about soil health or when they're reporting soil health? And so we found there was really wide variability in what depths people were sampling, how many soil samples were being collected. And so many studies were just collecting a single 1-inch thick soil sample right at the soil surface, whereas other studies were collecting large composite samples that could extend 3 feet down into the soil profile. And so when you try to make these comparisons across studies, it can be challenging if people aren't taking that same amount of soil, that same depth, and are not reporting the information in the same way. We also found that a majority of studies were just reporting data from a single sample, meaning they collected 1 soil sample and analyzed it for their soil health indicators. However, around 20% of studies collected and analyzed more than 4 unique depths. So again, there's quite wide variability in how people are sampling their soils when they're trying to quantify and talk about soil health.
So obviously you're trying to just start with what are people measuring and how are people measuring it. But the obvious next step is whether or not the things that they're measuring are actually useful or good indicators of soil health. So I imagine it's really tricky, though, to wrestle through that when you have people who are measuring things all different ways. So did you have any experiences with trying to kind of reconcile these different types of measurements as you were maybe looking at some of those next steps?
Yeah, absolutely. It's a big challenge when you try to do any kind of meta-analysis is how do you even approach data, because not every study is set up the same way. They're not measuring the same things. In this case, oftentimes they weren't even examining the same depths of soil. And so for our analyses, we had to basically link every measurement of a soil with cover crops to a corresponding control where there was no cover crops. And that allowed us to have these basically side-by-side comparisons of what was the value in the soil with and without cover crops. And so we use what's known as a response ratio, which basically is a ratio of the value in the cover crop versus the value in the control. And that allows us to basically take out some of that background differences due to different methodologies, different soil types, different crop systems, all of these other factors that really influence the data.
And that was where your 10% increase in soil health was coming in.
Right. It could be a 10% increase in the parameter, or in some of the parameters, we also said a 10% decrease. So an example could be the amount of erosion that happened in the field. Oftentimes when people do these studies, they see that once you put a cover crop in there, you see a decrease in the amount of erosion. So that'd be an example where you could have a 10% or greater decrease in erosion in those cover crop experimental plots or fields.
So what did you find? What traits were the most helpful to keep track of or not?
Right. So again, our criteria were pretty specific. We set a greater than 10% change in the response ratio due to cover crops, and we looked at only 1 to 3 years after implementation of cover crops. And so with those criteria, we found that 13 out of the 42 total indicators met this threshold. And so some of those indicators that we found were responsive that met this 10% response ratio threshold included soil aggregate stability, soil erosion rates, infiltration rates, and there probably is a relationship between those. If you have higher infiltration rates, you're likely to have less erosion. We also found some biological properties that were responsive, including the amount of carbon and nitrogen that are associated with microbes that are living in the soil.
And how did these traits split out across these 6 categories that you had? Did you find that one category had like a ton of them and one had none? Were they pretty evenly spread out? How did that shake out?
Yeah, so maybe a little bit surprising to me was only 2 of those indicators were considered, or at least we consider, to be physical indicators, even though when you look at what people are measuring, the most common indicators that people measure are physical properties. And so of those, we found that soil aggregate stability and infiltration rates were responsive. Actually, the categories with the most response were some of these biological indicators, like I said, the amount of nitrogen and carbon with microbes. Also, enzymatic assays that people have developed showed pretty good responsiveness, as well as the amount of fungi that were in the soil.
That's amazing. I mean, that's a huge thing to know that maybe what we're paying the most attention to isn't as helpful as we thought. So then obviously there's a few takeaways that we can get from this kind of research right off the bat. So can you talk about kind of maybe some suggestions that you would give people or takeaways that you would take from this research just right off the bat?
Right. So I think there are several implications from this work. And for one, just looking at what we found when we looked at what people measured related to soil health, there's not a lot of consistency right now with these measurements. And also when we looked at the responsiveness, at least over these relatively short time periods, a lot of what people were measuring and reporting didn't actually turn out to be responsive. And so here may be an analogy I would draw as to what's known as the McNamara fallacy. And basically the fallacies holds that we tend to really value things that we can measure and tend to discount things that we can't measure. In soil health, there are these measurements that we are doing that maybe aren't the best indications actually of soil health, and there could be aspects of soil health that we're not currently able to measure that we're kind of discounting or ignoring. So I think that's something that we want to be cognizant of. Are we measuring the right things? Are we measuring them for the right reasons?
Sure. And this might kind of transition into our next portion here about future research, but I imagine one of the things that you mentioned was also just we need to figure out what is the best way to report these and try and reach maybe more of a consensus on what kinds of things we're reporting. So there's more consistency there. As we talked about before, I mean, part of what makes this kind of project really difficult is just data that is coming from all different kinds of methods of reporting. You know, obviously you found tons of things that were being reported on. So I imagine that's a big portion of it too, right?
That is right. And I'll say that there is a lot of efforts, or a number of efforts that are currently underway to try to do some of this standardization. And so I've talked about the Soil Health Institute, but they released a list of what they called Tier 1 soil health indicators. There were 19 initial soil health indicators on there. And basically, to become a Tier 1 indicator, they had to meet some criteria. They had to be well-defined in terms of you could measure this parameter and know roughly what is a poor response or a good response or an adequate response. They have to show some responsiveness to management practices, such as cover crops. And so maybe it's interesting, in our analysis, we included all those 19 Tier 1 indicators, and we actually only found that 4 of them met our criteria in terms of the 10% response over the 1 to 3 year timescales. The Soil Health Institute's also released a list of Tier 2 indicators, And these are also ones that people have developed, but really don't have enough research or enough backing in them right now that we can interpret them or that we can consistently know this is a good value, this is a bad value, etc. The USDA Natural Resources Conservation Service also has a technical draft note out where they have put together a list of their own 6 recommended soil health indicators. And so I think this is encouraging that we're trying to standardize these sets of indicators. Hopefully that will encourage people to start being more consistent with what they report. But I also think it's important that we start thinking about standardizing how we collect our samples, right? So that, again, it's not some people are collecting 1 inch, some people are collecting 36 inches, because I think there is really the future is going to be in taking these results, putting them together, and start seeing kind of what are the common responses and how can we interpret these soil health indicators relative to the soil type, relative to the climate, relative to the management that's being done on those soils, because that's really how we're going to make use of soil health indicators. And as to the question of refining indicators, there's this whole list of the Tier 2 indicators that the Soil Health Institute has put out. And there's even Tier 3 indicators, which are even further away from prime time, you might say. And we still need more assessment on these indicators. They may be indicators ultimately that are really useful, that really help us dial in our management practices. And included in that, there are a lot of biological indicators that seem to hold promise in terms of being responsive to management practices. But some of them actually may be too responsive. They might change on the order of hours to days, just depending on what's going on around them. Still, I think there's a lot of promise in some of these measurements and methods. And with more research, I expect that they will become increasingly useful. Another area I think that there is and should continue to be emphasis is on some of the longer-term responses. And again, in our study, we really looked at that short-term response, 1 to 3 years. But right now, the Soil Health Institute is conducting a study where they're looking at long-term data sets. And so they basically went around the country, solicited people that had maintained a set of these soil health practices for more than 10 years, and then are prescribing a consistent set of soil health measurements so that hopefully they can learn what are some of the longer-term soil health benefits of practices like cover crops or conservation tillage. And so from my experience, I can say it's challenging to maintain research plots even for 2 or 3 years. So I really commend those people out there that are making the investments in time and resources to maintain these longer-term experiments. And I'm really excited to see what that effort reveals.
Yeah, I think this really gets down to the heart of why I'm so fascinated by this project, because it seems like so much of it is like just following lines of questioning, out to their final ends. So, how do we know that we are using the right keywords when we're using the research landscape tool? Or how do we know that it's the cover crops that are doing it or something else? As we start to find these more consistent ways to do the research or take the measurements, like how do we know that that's the best way to measure that, so it's just kind of this big ball of questions that just leads from one to another. And I love that there's so much potential for future research to really follow those lines out. So thank you for doing that research. But also on a more definite piece of progress, as we're moving forward with this, you guys are also working on a soil health calculator. Can you talk about that a little bit?
Yeah, so we've been working to develop a soil health calculator and really building on the results that went into this earlier analysis, where we looked again at cover crops. And this time we took away the constraint of the 1 to 3 year time scale. And we just basically compiled any study we could find that had to do with cover crops compared to a no cover crop control. And so we went through and basically looked at the responsiveness for all 42 of those soil health indicators that we found, and then focused on a subset that we felt were responsive based on our analysis or of interest to farmers, things like crop yield, weed pressures. And so basically, once we had all the data, we were able to break it up into different categories so that we could say, well, how do these responses change in different climates? And so you have the ability to select between tropical climates or arid climates or temperate climates or even continental climates, because it turns out that these soil health parameters respond differently in different climates. You might see decreases in certain parameters in an arid climate and increases in a temperate climate. We also were able to break down the data based on soil texture, if it's a sandy soil versus if it's more of a clay soil. We were able to break down by different cash crop rotations. So look at the difference of these indicators in a corn monoculture compared to a corn, wheat, soybean rotation. And we also were able to look at different types of cover crops even. So if you could have a grass cover crop in there, like a cereal rye or barley, and see how those indicators changed relative to if you grew a legume, which is this nitrogen-fixing plants like hairy vetch or clover in there. And so once we put all this data together, we were able to then make predictions of, on average, how much these different indicators are going to change once you add cover crops into your rotation. And so we hope this will, we think there's some scientific rigor behind this, obviously. We're doing these meta-analysis using all these data. We also want this to be a teaching tool and a learning tool as a way for farmers and regulators and practitioners to be able to at least see this is kind of what you expect on average based on all these studies that kind of have the same set of conditions as yours.
So is the purpose of the calculator then to punch in what you already have and see how things would change, or is it calculating some kind of percentage of how healthy your soil is, or is that maybe like a next step or a way to find gaps where you could be improving, or even pinpoint which of these indicators is the source of your problem if you're trying to, if you've got some kind of weird problem that you're not sure what's causing it, or how practically do you see people using this besides what you just mentioned?
I think there are a lot of really neat applications that we should be developing. Like you said, I'd like to work to you can put in your actual values and get an estimate of change. There are, of course, some limitations to an effort like this, that we're data limited. You're not going to have 100 different studies that all have the exact same set of conditions. So again, these are on average kind of results. Individual results are going to vary. But I think one of hopefully the big takeaways from a tool like this is right now, like you said, we know in the aggregate cover crops and these conservation agricultural practices are beneficial. They have environmental benefits. They oftentimes have agronomic benefits. But that's not necessarily guaranteed, particularly like on these short 1 to 3 year timescales. And so farmers make economic decisions. And so if they're not seeing a return immediately on extra expenses associated with cover croppings, this may be a way to say, okay, yes, this year you didn't see a yield increase, but looking across all these different studies, on average, you can expect to see a 10% or 15% yield increase through time.
That's amazing. That's very exciting. What a useful tool. So thanks for working on that too. And then also there's another, like we said, there's just tons of areas for growth in this field. No pun intended. So one of the other things is doing more meta-analysis and then also developing and refining these indicators or how we measure them. Can you talk more about that side of things?
I think if you look around, there's a lot of talk in various disciplines about big data. And so I think there's an opportunity to bring big data to soil health. And that's one of the reasons that we focus on some of these meta-analyses. It's a way to take information from all these really nicely done, controlled studies done in all these different locations and start seeing what are the emergent results from those. And so right now, we're working on another meta-analysis where we look at cover crops, and our specific question there is, how do they change soil organic carbon? So the amount of carbon that's there in the soil. And there are a lot of reasons that we're interested in this. Soils have been, particularly agricultural soils, have been proposed as this way that we can start sequestering atmospheric carbon. And so there's been a lot of studies that have gone and looked at under these particular set of conditions, this is how much cover crops changed or did not change the organic carbon levels. But using these meta-analysis, we've started to figure out things like soil organic carbon changes on average more quickly when you use mixtures of multiple legumes. So it could be a clover plus a pea together compared to using pure grasses like a cereal rye. And so I think there are all kinds of opportunity once we start standardizing our soil health measurements and the way we collect these data, that really there are almost infinite number of really important questions we can start asking of these data.
Sure. I've got 3 questions left for you. So the first one is, if people want to learn more about these projects, these meta-analysis, anything related to this, where can they go?
Great question. There are a number of resources out there. So the Tri-Society journals have been really publishing a lot of studies related to these conservation agricultural practices and soil health, including agricultural and environmental letters where we published our study. The Natural Resources Conservation Service has an entire website devoted to soil health. They have fact sheets, they have other information, they have graphics you could use, you could put up in your classroom, or you really could put anywhere. The Soil Health Institute's another place where you can find all these great resources like the Research Landscape Tool. And the Sustainable Agricultural Research and Education Group, otherwise known as SARE, also has a number of resources related to cover crops and soil health. For example, they just released a cover crops economics bulletin that basically broke down some of these economic reasons that producers would want to consider cover crops.
Second question for you is, if people want to get involved with this ongoing discussion or any of this research, what can they do?
Yeah, so there are a number of organizations, again, working on this question of soil health. And so an example would be the Soil Health Institute that has an annual conference that moves around the country. And the Soil Health Institute is sponsoring more focused meetings. An example was last October, there was a meeting in Silver Spring, Maryland, that examined connections between human health and soil health, and so brought together people from the fields of agronomy, environmental science, and even the medical fields, people that are working on the forefront of human health. It's also important, in my opinion, to continue to remind Congress of the importance of supporting agricultural and environmental research, these studies that are the backbone of our analyses and all of these other analyses that are possible with these data. Congressional members always like to hear what's important to their constituents, so writing letters and making phone calls is an appreciated way to just let them know that this work is important and valuable.
Last question is, what is one fun fact that people would not know about you if all they had was your research?
Okay, so soon after getting married, my wife and I decided to do a bicycle trip. We ended up riding bicycles from North Carolina to Oregon. It took us 2 months. We covered about 4,000 miles, and we really had many unforgettable opportunities to meet people across the country, including one woman in Kansas that we still write letters to. Still, that didn't seem like too much of a honeymoon, so a couple months later, we went to Hawaii for a week, and we didn't touch a bike the whole time we were there.
Yeah, I imagine after that, you maybe want a bit of a break, although I bet you guys were in great shape by that point, so you got that going for you. Wow, that's really cool that you still write letters to that woman. That's super neat.
And that's the royal we. My wife is the one writing the letters, but...
Oh, okay. Well, still, I'm glad that you guys met a new and lasting friend. How wonderful.
Yeah.
So, yeah, thanks for being on the show. Thank you for lending us your expertise as we wrestle through such naughty definitions as far as what soil health is and how we measure it. Best wishes on your future research, and thanks for being on the show.
Thank you very much.
Thank you for listening to Field Lab Earth. You'll find a link to today's paper and other resources for this episode in our show notes or on our website. If you have any questions, comments, or recommendations for show topics, please contact us at podcast@sciencesocieties.org or on Twitter at FieldLabEarth. If you'd like to hear more content like this, please subscribe, and don't forget to rate and review us on Apple Podcasts or anywhere else you find your podcast if you like our show. This podcast is a joint production of the American Society of Agronomy, Crop Science Society of America, and Soil Science Society of America. Special thanks to Lobo Loco for the use of their song Spook Castle on the intro and outro of our show. Opinions and conclusions expressed by authors are their own and are not considered as those of the American Society of Agronomy, Crop Science Society of America, Soil Science Society of America, its staff, its members, or its advertisers.